Hello, this is John Wecker reporting for Life Science TV. I'm here at Life Science Innovation Northwest 2016, and I'm joined today by Dr. Margaret McCormick. Margaret, thank you very much for joining us.
Thank you, John.
And congratulations on your award, Women in Life Science.
Thank you.
Well done. So I'm going to just let the audience know that we have a relationship, as it were, outside of work. When I joined the board of Life Science Washington, you were the board treasurer, and I got to know you through that experience, and I developed a really deep respect for your willingness to ask the tough questions and attention to detail, and just it was clear you were a very thoughtful, conscientious person. And as a result, I thought you'd make a great addition to my board at the Pacific Northwest Diabetes Research Institute, and I thank you very much for joining us. And I look forward to having you ask me the tough questions from now on. So thank you very much. Oh, good. So you're the CEO of Matrix Genetics, is that right?
Correct.
So tell us a little bit about Matrix Genetics and what's really exciting, what's going on there that's really exciting.
Great. So Matrix Genetics is an industrial biotechnology company. So we're outside of the cure side of biotech and really focused on the fuel, feed, and food side. And so we are a synthetic biology company utilizing photosynthetic microorganisms to produce really cool products. And so right now we've got a number of different programs. Our largest collaboration is with a major energy company that has dedicated its efforts to trying to find renewable fuel sources. And so we're their advanced biology arm.
Wow.
About 20 of our scientists are actually working on that project.
Wow.
And then on the other side, what the exciting development we've had in the last couple of years is that we're the first and only company or lab actually to figure out how to genetically engineer spirulina, which is already considered food. It has what's generally recognized as safe or GRAS status. And so by taking this very high protein food and trying to make it better, there's so many different ways we can exploit that technology. And so that's what we're really focused on right now.
Wow. So energy and food production. What other areas?
So the types of things we can do is develop pigments. If you think about just globally right now, there's a huge push to get away from any type of artificial dyes. And the color blue is one of those pretty interesting things where there's not a lot of blue in nature for whatever reason. But blue-green algae produce it naturally. And so if to get a natural blue hue that's a true blue, not like a reddish purpley blue, you actually have to go to an organism like spirulina. So we've got programs around developing that type of pigment, which would be for anything from Gatorade to ice cream. But it's also used as a medical imaging dye, so it has other types of uses as well. But then taking that whole idea that we can take something that's used as food and make it better, we're really looking at the enhanced nutrition space. And not just focusing on human markets, but also livestock and aquaculture markets.
Wow.
So one of the programs that we've got going right now, which is really exciting, is can we make this spirulina and actually make it an oral vaccine? And an oral vaccine for aquaculture. Because looking down the road, by 2030, over 60% of all fish consumption globally will be through farmed fish. And so one of the big issues with these types of farming operations is culture crashes because of disease. So we're looking at novel ways that we can protect the fish and then go on and look at other types of livestock by incorporating into the feed as opposed to going through the typical injection process.
Wow, that is really some exciting work. So how did you get there? So tell us a little bit about your career path and how you ended up in Matrix Genetics.
Sure. So when I went to grad school, I ended up in a laboratory that was not focused on the typical disease models or basic biology, but it was a very applied biology lab. And there were two projects. One was making bioplastics in bacteria, and the other was making amino acids in bacteria. And so I was on the amino acid side. So our collaborator at the time was Archer Daniels Midland, because things like lysine, threonine, those are huge markets also in the feed area. And so my graduate life was focused on that.
And was this in Wisconsin or was Baylor?
No, this was actually at MIT.
At MIT, that's right. I
Yeah.
forgot about it. Yes.
So after grad school, though, I decided to go to the business side of just the business side. I was recruited by a company called McKinsey Company.
Ah, yes.
And so ended up in their Houston office where I ended up doing a lot of energy consulting.
Okay.
Anything from retail strategies to lignite mining. And after a couple of years of that, I was like, well, let me go back to life science where it's where my passion was. So I ended up at Baylor College of Medicine.
There you go.
And this was a great organization within Baylor called BCM Technologies. And there was a tech transfer arm of BCMT, but there was also a venture startup arm of BCMT. So while there, I worked on 8 startup companies, helping to get them launched. Baylor had set aside $20 million to invest in these companies. So we found, with the Baylor investment and then co-investments, we did a great job of getting these different types of companies up and out. And eventually, I spun out to run one of them, which was on the drug development side. Fast forward a couple of years, my husband got a position up here in Seattle, and we decided to move, which was a great decision. And I ended up with a life science venture capital firm and called Integra Ventures and led many of their biotech investments before eventually going to one of the company's Targeted Growth.
Okay.
And which is an agricultural biotechnology company. And within TGI, Targeted Growth, we eventually spun out Matrix. And so that's...
There's your story. Okay.
It's a very circuitous route.
I want to go back to that moment in time when you made that transition from an academic to a venture capitalist or a consultant, a management consultant. How did that occur? What transpired there?
So I think I decided that I didn't want to be an inch wide and a mile deep anymore. I loved learning, and I loved learning a lot of different things. And I saw a future where, as you became to be a fantastic scientist, you really need to be focused on a fairly narrow area of science. But I liked being broader than that, and I liked learning outside the science area. So I saw going to McKinsey as a way to basically broaden my options, and it turned out to be true, because even though I didn't end up, pursuing a career at McKinsey, getting that business experience was very helpful. And then I've been able to apply both the business side as well as the technology side throughout my career, which I think is hugely beneficial.
No, it's hugely beneficial. So if you look back on the many, many years, the few years.
Just many.
Just many, okay, just one many. What's your greatest satisfaction? What really feels fulfilling when you look back?
So, I've really focused on the startup side of the business, and both on the operations as well as on the investment side. And I think, looking back, that a lot of the companies have made tremendous progress, both getting deals done, getting products to market. But it's really about the impact on other people. So, hundreds of jobs have been created because of the work that I've been directly involved with and indirectly involved with. And that's great, to be able to give other people the chance to really pursue their dreams, to pursue their professional opportunities, and, to see them make impact, I think has been one of the most satisfying things.
Good. So, women in life science. And I've had a couple conversations with your fellow awardees today, and the question often comes up, why do we even need to continue to have a special award? Why isn't this just normal business? We're not where we'd all like to be. But if you had some advice for especially, maybe we shouldn't be women. Anybody in a situation where diversity is an issue, whether it's race or gender or whatever, what would be your general advice as they think about a future in science or a future such as yours?
So the way I've always looked at it is it really comes from within. You have to make the decision to overcome whatever obstacles there are. And there are obstacles, but you have to find what you're passionate about and pursue it. You have to identify your strengths and exploit them. Also identify your weaknesses and figure out a way to mitigate or overcome them. And then, most importantly, you really need to step up, step in, and lead. And if you're afraid to do that, you're probably not going to make it. Yeah.
No, in many ways, it boils down to that you really need to know yourself. As you did when you made that transition. You knew where you were at that time in your academic career, and you knew that wasn't the direction for you. So,
yeah, well, it's I knew early on that, I wanted to be successful.
Yeah.
And I was super shy, like going through elementary school.
Really?
Oh, yeah, like super, super shy.
Okay.
But I realized that if I wanted to make it, I had to figure out a way to overcome that.
Yeah.
So, you know, if you want it badly enough, you can do it.
Good. I think that's great advice, and I
Great.
look forward to working with you in the future. And good luck with Matrix Genetics, too.
Okay, great. Thank you, John.
So for John Wecker, I'm John Wecker, and this is reporting for Live Sciences TV. All right, everybody, bye-bye.